Namaskar, my dear friend. I once again brought a very important video, working function of the SEM EDXA, scanning electron microscope, energy dispersive X-ray analyzer. In forensic laboratory, SEM EDXA very important equipment used for analysis of different kind of the samples. It may be used in the physics division, ballistic division, chemistry division, biology, etc. as per our requirement. And the type of the samples is not limited, but it is mainly used for analysis of the two types of the analysis. Number one, physical or the topical features analysis. Second is elemental composition analysis. The component, the element component of the substance. Hence, we are going to analyze of a sample. We have to know the working function of the equipment, like in this case SEM-EDX. So in the short video, I have tried to explain the working function of this scanning electron microscope. So in a scanning electron microscope, this is made of three parts: SEM, EDXA, and compute section. So in detail, here, so what is the parts? First, electron gun. The electron gun are used to generate the electrons. It is a special type of the electron gun. Different type of the electronic source is available, like a very simple first source is a tungsten, which are heated by giving the electricity coil. Otherwise, the other type of the electronic source also available. So this electron gun generate the electron, emit the electron, and this electron come in this direction. So for increasing the energy of this electron, as per our requirement, there is a anode. So this anode is accelerate the electrons. So the electron gain the energy, sufficient energy, which we are required. Suppose energy is a very less, of this fall the samples, IXA will not generate. Some energy is required. Essential energy is required. A threshold energy is required. So this anode applying the high voltage. In this anode, we applied the high voltage, like 20 kV, 20 kilo electron volt. 20 kV is applied. This can be vary. For this, for reference, I have seen that like the 20 kilo electron volt. So as per requirement, the voltage can be applied in this anode to give the sufficient energy to the electron. And now this electron comes in another lenses, which is called a condensed lens, because the electron is maybe spirit. So for going the condense a particular direction, fixed direction, which given the not scattered, the condensed lens are used. So these lenses are not a physical lens. This is an electromagnetic lens, a condensed lens or a objective lens. These are not physical lens. They are in this SEM, the physical lens made by the glass or any material is not used. Is the magnetic lens, electromagnetic lens. I will discuss in the next video how they work, but keep in mind here the lens is not a physical, like a glass or any other material. They are made by a arrangement, special arrangement by the coils to create a magnetic field. So this is a electromagnetic lens. So both are lenses are the electromagnetic lens, not a physical lens. And there the samples falls on the sample. And these samples are put a special stage. This is a sample stage. See. And this stage can be traveled this direction, as per requirement, because X-ray is coming in this direction, our sample is here. So this should be in the spot of a X-ray. X-ray should be fall on the sample. So this can be moved at 360 degree. This direction, even the tilted. So any direction 360 degree can be moved. Yeah, this motorized stage. And here is a sample. So the electron, this electron falls on this samples. And different type of the action is take place. Like some X-ray is generated with the interaction of the electron with the material. And this X-rays is detected. And the nature of X-rays is a characteristics feature of the samples. So elemental analysis is detected by the analyzing of the X-ray detector. Further, the computer can detect the different. So how they analyze the elemental analysis, I will discuss in the next video. What the X-ray generated with the interaction of the electron with the sample is a characteristic feature of every samples. Every different types of samples give the different type of X-rays. The property of the X-rays, their wavelength, their energy is different. So by analyzing the nature of this X-ray using a separate detector, the elemental analysis can be determined. And another, when this fall secondary electron, they generate also the back scattered electron and secondary electron. And this secondary electron or back scattered electron are used for the image formation. So the image is formed, but this image is very large. So the tropical features, the tropical structure, which are the structure are present on this surface of the material can be analyzed. So two types of the analysis is being done by the skin electron microscope. Element analysis. Number element analysis. Element analysis means using the X-ray detector. And the second is a tropical analysis. Surface structure. This is a secondary electron or the back scattered electron. They form the energy. Secondary electron or the back scattered electron. So this is a short video. I try to explain the working function of the skin electron microscope. If you think this is useful, please subscribe my channel. You can contact also at this number. Thank you.